episode of Progress, Potential, and Possibilities, discussions with fascinating people designing a better tomorrow for all of us. I'm your host, Ira Pastor. Welcome, everybody, again to another episode, our actual 2022 inaugural episode of Progress, Potential, and Possibilities. And we're going to be starting the year off with a bang with another really fascinating guest for everybody who's helping to indeed create a better tomorrow. And in an area which, as many people that watch the show, I'm very excited and passionate about myself, that is regenerative biology. And we have the honor of being joined today by Dr. Maximina, who is research group leader at the Center for Regenerative Therapies in Dresden, at the Technical University of Dresden, jointly affiliated with the Max Planck Institute for Molecular Cell Biology and Genetics. Dr. Yoon and her group study the cellular and molecular bases of regeneration of complex structures with the help of organizations like salamanders, including newts and I'm pronouncing that wrong, waxadiles. These vertebrates have, as we discussed in previous episodes, really exceptional regenerative abilities, which in contrast to humans are complex, capable of regenerating very complex tissues, even entire organs, to a quite remarkable extent. And hence they offer very unique insight into what molecular mechanisms ultimately must be in place for achieving such perfect regeneration. Research in the Yoon group focuses on three main areas. One, in describing the various cellular and molecular mechanisms that help drive regeneration, or mechanisms underlying the plasticity of cells, the connection of regeneration to cellular aging and the role and regulation of senescence in regeneration, and lastly, the role that the immune system has in the regenerative context. The research in the Yoon group combines advanced molecular biology methods with state-of-the-art microscopy. Most recently, the group has established the Salamander-Eci, a novel method that enables three-dimensional visualization of salamander tissues for a comprehensive understanding of regenerative processes. Dr. Yoon did her Ph.D. in biological sciences, MRC-Laboratory of Molecular Biology at Cambridge University. Her postdoc research in the Institute of Structural and Molecular Biology, University College London. She did her biologic science bachelor's, University of Buenos Aires in Argentina. A lot of exciting things to discuss today, but Dr. Max Yoon, thank you so much for taking the time to come on the show today.
Thank you, Ira, for having me.
It's great to have you. I've been looking forward to this episode for a couple of years now, but I would love to start things off as we typically do on our show by handing you the floor just for a little bit to talk more about yourself, everything from where you grew up, when you got interested in biology, how you got interested in this fascinating area of regenerative biology, and then a little bit of the early career journey. I think that'd be a great way to start things off.
Sure. So my beginnings in biology were in Argentina, in Buenos Aires. So I attended the University of Buenos Aires and I specialized in molecular biology. And very early on, I would say, when I started my undergrad, almost straight away, I started to work for free, as one did at the time, at a very prestigious institute in Buenos Aires. And that allowed me to have laboratory experience almost all the years where I was completing my studies. And that was great. It was in the topic of plant pathogen interactions, but it allowed me to first get an introduction in genetics, because I was building mutants for different bacteria strains of Xanthomonas, which is an important pathogen of plants. And different countries have different stresses in the type of sciences
Sure.
that fund, and Argentina has a big sort of a strong investment on sciences in the area of biotechnology, agrobiotechnology especially. So this kind of made sense. And not only that, but it allowed me to get already into a complex biological system, because you have plant cells interacting with bacteria and looking at the responses both from the bacterial perspective as well as from the plant perspective. And the bacteria triggers a response which is a bit akin to an immune response in the plant. So there's a local response, for example, callus deposition to restrain the bacterial growth inside the leaf, for example, as well as the production of signals that alert the plant that is being under threat by a pathogen. So it's a little bit like the immune system of the plant, and it kind of connects to what I'm doing now. So after graduating from university, I had the chance to go to the very famous LMB at Cambridge to do molecular biology again and genetics for my PhD. And there I focused on mutations, but this time in vertebrate systems in order to understand the basis for genome stability maintenance. So I went into looking at the mechanisms of homologous recombination then, and how the DNA is able to be repaired. And while I was continuing these studies, I got an offer from Emmanuel College to start to do supervisions in cell and developmental biology, which is an undergrad subject. And maybe some of your viewers are aware, so the Cambridge and Oxford systems are particular because undergraduate students that go there have the remarkable luck and fortune to be mentor and tutor on the particular subjects by specialists, either reputable scientists or students who are very keen on the topic, besides the lectures. And this involved me as a young student then getting very involved with the topic of cell and developmental biology, especially, which wasn't part of my background. And as a key topic in developmental biology, you have to teach also regeneration. So that was kind of my first, serendipitous encounter with regeneration, if anything. And I was fascinated by it. So I then decided that for my postdoctoral studies and perhaps my future career, I wanted to really get into regeneration. Obviously, when you are at this junction, you have to consider, who could be a good mentor in the topic? And by then, I looked around and there were a number of great scientists. Then you combine the mentor with the model that you think is more appropriate to study regeneration. And for me, that was a no-brainer because I was fascinated straight away by the power of salamanders to regenerate a number of complex structures, and the fact that they are the closest vertebrate to us that is capable of all these wonderful regenerative feats, right?
Yep.
Even closer than the zebrafish. So they are truly the closest organisms to humans that can regenerate organs, entire organs, for example, or entire structures such as their limbs. So that was kind of a key moment in my scientific career. And then I joined Jeremy Brox's lab and stayed there, for my entire postdoc, which was about 6 years, I would say. And they were wonderful because the kind of mentorship I received, the knowledge, the know-how that was already in the lab really helped me for my future career and to eventually develop and expand some of the ideas that we were working on then, and develop some new research lines that we're very much pursuing now. So that was a very great moment, I would say, in my career.
Absolutely. It sounds like it. I'm just thinking this through, and yeah, when you combine sort of the knowledge base plus the mentor, yeah, I mean it seems like extremely opportune for you to come along and now really be leading the frontier in this space in 2022. So yeah, it's a very exciting intro story. Max, as I'm sitting here listening to it and thinking about it, and we chatted a little bit before the show of sort of my own personal interest in this space. The field of regenerative biology is really interesting in the respect that, and we'll go into sort of the details of your research in a little bit. But when we think about that, it goes back quite a bit. Some of these books, so far, I have a few of them here, one by Bruce Carlson, who I had the opportunity to talk to a couple of years ago, University of Michigan. And I go through some of these textbooks and I'm looking at the references, and they go back, about 100 years or so, some of them, to these really sort of complex studies of morphogenesis and regenerating structures and a variety of species. And then we sort of come to an era, as you know, in sort of the mid 20th century, where things went in the direction of molecular biology and looking at genes and certain proteins and so forth. Very great. At the same time, regeneration, as you know, is a lot more than some of those genes. There are really complex dynamics going on in the biophysical component in some of these cases, and sort of the mysteries of morphogenetics and morphodynamics. Talk a little bit, as you've studied the area and become sort of interested in it and amazed by it, as you look back on those 100 years, sort of where have we come, and sort of where do you think we're going in terms of how we can remarry a lot of what you're doing at the molecular and cellular level with some of the sort of the higher level dynamics, the organs, the tissues, the more complex things that might not have a specific target per se, but are a very important part of the complexity that regeneration is?
Great. So this is a great and very complex question.
Sure. Run with it any direction you want.
But I would like to start first to tell you a little bit about the history of regeneration,
please,
as you mentioned, and how actually some of the topics that were already defined in the 17th, 18th, 19th century are still very much active today. So, I mean, they are still very relevant and we keep going back to them. So obviously, if you want to think, historically, you have to start probably with Aristotle, because Aristotle basically described already in his Bestiaries the phenomenon of regeneration. In particular for salamanders, one of the first and best descriptions is with Lazarus Spallanzani, and it's already in the 1700s, with a very sort of detailed account of tail regeneration, of limb regeneration already. But one of my favorites was a collection of essays, I will call it book, is Regeneration by Thomas Morgan.
Okay.
So Thomas Morgan is famous for his work on Drosophila and genetics. And he is widely recognized for it, but he, even before that work on Drosophila, he was very interested in regeneration, and he published a collection of essays and lectures, like exposing his ideas on this topic, and it's already sort of 1901, I believe, so long time ago. But he poses, starts to phrase the questions that we are very much working on. Like, he highlights the importance of the regeneration modes, and that there are different regeneration modes. At the time, like, he was working very much with Hydra, which is this polyp that can regenerate very much, whole body wise and from different parts, right? And Hydra can exhibit sort of two types of regeneration. You can regenerate through activation of proliferation of progenitors and subsequent differentiation to give you the structure, or, and this is called epimorphosis by Morgan, or by simply, once it gets amputated somehow, it can reshape and restructure itself to form the new individual or the missing part, and this is called morphallaxis by him. So he already defines there's an importance in terms of the regeneration modes. They may not be the same. And this is something that the more we know about the organisms that regenerate nowadays, the more we find that there are different ways of regenerating. Another important concept is basically is something that is very simple, but at the beginning of the history of regeneration research, it was not very much investigated fully, is that a regeneration comes with the physiological restoration of function. And nowadays, because we have all the tools, we can address this. Of course, for something like a limb, it's a simple concept, right? You can see whether the limb is functional or not. For other structures, you're not sure, right? You have to then go and test it properly. For example, when an organism regenerates a part of the nervous system, you have to test for functionality. Again, it's a very important concept that in many systems, even though it's a simple idea, was thoroughly understudied. And then he also defines the impact of external factors in regeneration, which we know is very much true, such as temperature, gravity, and many others, and internal factors, which of course we now have learned, and also through scientists, even sort of contemporary to Morgan, are very important. And there's a number of cellular systems which we know are crucial for regeneration, and this involves nerves. So this was first reported by Tweedy Todd in the 1800s, but there is no regeneration without nerve signals. And this makes sense, right? Because you don't want to regenerate a structure that eventually you cannot control. But there's this marriage between regeneration and nerves. And there's another very important concept which also was recognized by Morgan, which is the importance of the tissues that remain once a structure is lost. And this is very important, we know, because they have a double function. They need to provide the progenitors for the regenerating structure, but they also have to instruct somehow the identity. I mean, it's not the same when you lose a hand or when you lose an entire arm, right? So the tissues that remain at the amputation plane somehow know what their position or their identity is within the structure. And therefore they know how much to regenerate, right? And what to regenerate. So these are very important concepts that guided regeneration research up to now. I would say, for example, the field of regeneration is currently very active on understanding the mechanisms that underlie nerve dependence, and very much active on the mechanisms that regulate the establishment of positional identity, which is crucial for regeneration. And it's remarkable, but this was already written down in 1901, and here we are, 2022 already, and we still don't know the full answers, and for some, not the answers at all, right? So that's kind of very, very interesting. Now, of course, through the time, like you can incorporate other important concepts in regeneration that some of them are almost universal in these systems that regenerate. For example, we saw the importance of establishing a wound epithelium, which is a specialized epithelium that covers the regenerating structure and sends signals to the regenerating structure in terms of growth and patterning, for example. And we know that that is special. It's not just like simple skin, because if you're looking at regeneration of a limb and you replace that wound epithelium for normal skin, then regeneration will stop. So it's a very important structure, this wound epithelium. Now, eventually we learn much more about other cellular components, such as the immune system, and how the immune system is actually required to promote regeneration in many contexts. And we've seen this in salamanders, but also in xenopus, in zebrafish. Perhaps the components or the signals that they send are maybe slightly different or toned down in different systems. But the importance of these players, for example, especially macrophages, I would say, is there and is evident. And then, with time, scientists started to pay attention to other more complex signaling systems, as you mentioned. For example, electrical signals. We know that upon the loss of a structure such as a limb, there are a lot of changes in ionic currents in the structure that are important for regeneration per se, because if you revert, for example, the polarity of those currents, regeneration may not occur. So, and these are early experiments already in the 1900s that were very, very significant. And as you mentioned earlier, so you had Michael Levin here, and he works pretty much on that area and trying to figure out the importance of electrical signaling in these type of events. Now, we are also now seeing, with the development of the new tools as well, the emergence of a new side of regenerative biology, which focuses on the mechanical aspects and how mechanics influences how a tissue regenerates. Is it instructive or not? And is it instructive for growth, for patterning, for cell plasticity? So this is another area that we've seen has emerged since then. And on a more recent trend, you can also mention metabolism, which, of course, now with the with the available tools on metabolomics and lipidomics, one can actually track and start to look a bit more in depth into the influences of metabolism in regeneration. But coming back to the integration of a phenomenon that is very, very complex, as you mentioned, one important consideration here to make is, of course, the availability of both transgenesis and single cell technologies are really starting to allow the understanding of cellular cell-cell interactions and coordinated, basically, interactions within the tissue structure. Because you can have snapshots, especially with single-cell technologies, of the transcription programs that are active in each of the cells that is regenerating. And if you can do time lapses of this and build trajectories, then you can try to understand how these cells are relating to each other, which signals are producing, what signals are receiving, and then try to build a better picture. That then guides, obviously, functional studies. So I hope this gives you a little bit of a panorama.
Yeah, I'm just, I'm enthralled by your sort of encyclopedic knowledge of it all, and really, just as I said, before the show, just that someone like you, here we brought Aristotle in, so we have a few thousand years here of history. I'm glad to see that you are going to be the face that's leading the next next generation of this. So this is really exciting, and yes, thank you for that overview. I wanted to, take some time now and to allow you to go sort of deeper into some of these components, specifically the ones that you're focusing on in your lab. And obviously, one that's sort of gotten a lot of press lately in sort of biotech in general, sort of this concept of cellular reprogramming, dedifferentiation, and sort of the ability to move the plasticity of a cellular state. Obviously, I guess this theme with the sort of the Yamanaka Gurdon Nobel Prize from a few years ago has sort of increased awareness of induced pluripotency. But, you know, this sort of cellular flexibility, as you know, is something that, you know, the salamanders of the world and the newts and so forth have been perfecting for hundreds of millions of years. And it's not just a case of pluripotency, but in many of, you know, I looked at some of your papers and in the area, there's really a very complex sort of cellular milieu, as you talked about sort of the marriage of many of these tissues. You have published on the involvement of things like P53, mitogen activated protein kinase, a lot of really complex molecular factors in this cellular plasticity component. Talk about some of what you're learning nowadays and sort of some of the directions that you're going as you study, and we'll bring up the word blastema into this, of sort of the cellular mix that's involved when we lose that part of the body or the degeneration of an organ. Talk a little bit about some of the things that you're focusing on specifically in terms of cellular plasticity.
So, I mean, as you mentioned, I mean, salamanders are masters of regulating cell plasticity. Yep. And it is true. I mean, as you say, there's now a big interest in reprogramming, in turning cells, you know, to their origins in order to perform subsequent, you know, differentiation into particular tissues, which is all great. But when it happens within the blastema and in nature, what you have is a limited reprogramming.
Got it.
So the cells go back to some extent, but not to the point that they can give you all cell types.
Got it.
And a good example for this is the newt muscle, which is a structure in which we have worked on extensively and we are still working on, and I'll tell you in a few seconds. So for those that don't know about the system, so salamander muscle can be grown in culture. Mm-hmm. And there, what you can see is that by giving some sort of serum stimulant, you can promote cell cycle reentry and dedifferentiation of these muscle cells. And this recapitulates very well what happens to this muscle when it's inside the animal. So inside the animal, you can label the muscle genetically, or, you know, or even implant muscles, which you can label outside the salamander, put them back, and then during regeneration, you can see what is their behavior. And what you can see is that muscle, during the regeneration process, it undergoes differentiation up to forming mononucleates. So the muscle starts to re-enter the cell cycle, it breaks out into little bits, and then is able to give progenitors. So those mononucleates, which come from the differentiated adult muscle, are able to enter proliferation and give you more of the same, and eventually differentiate again into muscle. So muscle during regeneration gives you muscle. But the important thing is, like, this is adult muscle, differentiated, is the one that is already in a fiber, which is very remarkable, because in most systems, for example, in humans, we regenerate the muscle when we do exercise or, you know, have an injury from satellite stem cells. So we don't do it from the adult differentiated fiber. So this is a big difference. And the newts are known for doing this very well and use it as a key regeneration mechanism. The same goes for connective tissue. So connective tissue during the regeneration, and we know this from single cell sequencing studies, it goes back to a point where it can give you more of the different types of connective tissue. But it's not going to give you muscle or neurons or Schwann cells, right? Sure. So each tissue goes back and forms more of the same tissue. It has a memory of what it is, as well as the position, right? Which is a very important topic. Mm-hmm. So since we figure out that For example, special regulation of p53 is required for the correct dedifferentiation of muscle, and this involves ERK signaling, as you mentioned. We kept using this system, and we're now doing something that is very, very exciting, I would say, which relates to a very recent finding of us, in which we reported that during regeneration of the limb in salamanders, this happens both in newts and axolotls, there is an induction of senescent cells.
Mm-hmm.
And these senescent cells eventually are cleared by the immune system and disappear, so they don't remain. Now, senescent cells, for those who are hearing this, and this may ring a bell but don't know exactly, are cells that are induced through the activation of a program upon a number of stimuli. And this usually includes cellular stresses such as DNA damage, UV light, activation of oncogenes, but are also known for being induced during physiological conditions such as development. Mm-hmm. And the characteristics of a senescent cell is that it stops dividing, it undergoes a permanent cell cycle arrest, and it starts to produce a number of molecules, which we call the senescence associated secretory phenotype. And these molecules involve a number of chemokines and remodelers of the matrix and growth factors, and together they are able to modify the microenvironment of the cell that is producing all these factors. Now, because of that, they could have various effects in a number of processes. So we actually started to look into whether senescent cells could participate actively in the process of regeneration and could be beneficial for it, could be doing something good for regeneration, mm-hmm. and eventually they are clear and they're gone. So one of our interesting findings, which is now very much pursued by one of my postdocs, Hannah Walters, so is that senescent cells can promote the plasticity of these mitochondria, can by themselves, through these secreted factors, promote cell cycle reentry in myotubes, even in the absence of these serum stimuli that we usually give ex vivo. Mm. And we have also seen that senescent cells within the limb can promote regeneration, can enhance the formation of epiblastema, and they can also induce cell cycle reentry of myofibers by themselves. So this suggests that the senescent cells are actually promoting the plasticity of the muscle in order to generate these progenitors for regeneration. So that's a very active sort of line that we are pursuing in the lab. And we have another very interesting line which refers, at least indirectly, to cellular plasticity, okay. which looks into how positional identity is established. Okay. So I mentioned before, positional identity is essential for determining how much and what to regenerate. So it's the information or the coordinates that tell the cells at any point along a three-dimensional axis where they are located. So it's essential for regeneration. And the problem we had in the field is that there are very, very few molecules associated to positional identity. We know a lot about the gradients, the morphogenics that establish the axis of the limbs, right? And that's from traditional developmental biology, we know a lot of that. However, the specific molecules that form the coordinates or that are involved in cell-to-cell interactions that tell one cell, I'm proximal, and the other cell, I'm distal. Mm-hmm. We don't know anything about that, or very, very little. So one of the very important discoveries from my postdoctoral mentor, Jeremy Brooks, was a protein called PROD1. And PROD is already telling you it's going to have something to do with the proximal axis. which has been proposed to determine proximal identity within a limb. And actually, when you give cells extra doses of this protein, PROT1, you can, transform them, or at least you will find them in more proximal locations after the regeneration process is done. So following, building a little bit on that, we found another protein called TIG1, which is, and we have a bioarchive paper already out, so I feel free to talk about this. So, and this is a protein that fulfills some of the criteria that you would expect for a determinant of proximal identity. So you would expect that that protein will have a gradient or exist in a gradient from proximal to distal, and this is the case. And you would expect it to be upregulated during regeneration at the time when the limb is establishing proximal identities, which we find. You would expect this protein perhaps to be able to reprogram cells when these identities are being re-established during regeneration, to proximal identities. And indeed, what we found is like, even if you give TIG1 to cells which are distal, when you give enough of TIG1 to these cells, you reprogram them to a proximal identity. And these cells end up in proximal locations, but also we look at the transcriptome of these cells. And these cells, first we label them genetically. We label the distal cells genetically. So the distal cells, we know because we are overexpressing a fluorescent protein driven by the promoter of HOXA13, which is a distal marker. So genetically, these are salamanders which are genetically modified. They have a knock-in HOXA13 driven fluorescent cherry, in this case, which labels all the distal domains as they are formed. So the domain that is going to give you your fingers, for example, your hand and your fingers, and it's labeling all in red. You can see it under the microscope. Now, you can then give a virus, for example, deliver DNA through a virus that gives these red cells, distal cells, the ability to overexpress TIG1. And when these distal cells start to produce TIG1, you look at the transcriptome of cells which are just distal, they are only red, or cells that are distal plus TIG1, and the distal plus TIG1, they are no longer distal, they are now proximal. So this really tells you that TIG1 is somehow able to control the whole expression pattern in those distal cells and transform them into proximal. So this is a very, very, very insightful lead because we don't have that many into how proximal distal identities are established. And we were very excited when we realized what was going on and that TIG1 had such a strong effect as to be able to reprogram a distal cell to a proximal identity. So we are now continuing with that research and trying to figure out how is TIG1 linking these coordinates and the expression. Because, we think basically different levels of TIG1 may result in different transcription patterns which are associated with proximal or distal according to the level of TIG1. But how exactly that is established is what we are trying to figure out right now.
Very exciting. Very exciting. And as you were talking about the senescence associated the secretory phenotype, You guys saw a couple months ago, had Judy Campisi on the show, and she was talking about these different SASPs. You understand sort of SASP, but we don't know if in aging whether a liver SASP may be different from a spinal cord, so whatever. There may be some differences here. And as you were talking, we have cytokines and chemokines and immunomodulators and so forth. I guess it's a little too early for this, but do you think that thinking about regeneration and some of these patterning dynamics, proximal, distal, and a lot of these unique, what I'll call regenerative law, that has been studied for so long. Do you believe that we're going to have to go into, with study, okay, if we pull the salamander's eyeball out, that there's a different SASP required for positional identity, obviously in the eyeball, than there will be in sort of the heart or the other, a limb somewhere. I know it's very early days on all this, but are you
I fully understand your question, and it is very important that this is something that we are seeing with all different kinds of senescence. So senescence cells can be generated from any cell type and in many conditions, because it's like apoptosis. Right?
Yep.
It's a natural response, it's a natural program, which is in principle anti-tumorigenesis, control, right? So either, if you have too much damage or too much stress as a cell, you don't want to pass that damage to your progeny,
yep.
or you don't want to engage in processes you can't. And you either stop your cell cycle to repair that damage, but if the damage cannot be repaired, then you undergo apoptosis or you trigger senescence. And that response, that capacity, is found in most cell types. Now, according to the cell type that enters senescence, you will have most likely a different SASP and a different response. And we are starting to see that. But even among the particular senescent cells that are induced during regeneration from a different cell type, and we started to do single cell analysis, single cell RNA-seq analysis, for example, of these cells. We know that it is heterogeneous, the population. And even if you can define a common SASP among a particular cell type, it evolves in time. So it's not the same, the SASP at, a week after entering senescence from the SASP that you have after a month after entering senescence.
Interesting.
And so this makes things all the more complex.
Sure. As if they're not complex enough.
Yes, exactly. Yeah. One of the emerging themes from all the data sets that we are now generating with the high throughput technologies such as multi-omics, right? So you have RNA-seq, especially at the single cell level, transcriptomic, ATAC-seq at the single cell level, is that we are seeing the heterogeneity of the system. Things that before you thought, okay, all these senescent cells are induced at this time, they're all secreting these factors, they are all doing this. Well, probably not true. Probably there's some level of noise in the system. Probably there's some level of specification. Perhaps it's just a proportion of those cells that you require for producing all the compounds. So we're seeing heterogeneity, and this is a general, as I say, a general theme that is emerging from now our ability to look exactly not only what the regeneration is how the regeneration is happening at the cellular level, with imaging, but also at the individual cell, single cell analysis. And it is heterogeneous, and there is noise, and we have to live with it because the process works like that.
Yep, that's biology.
Exactly, that's biology. Yeah. But one of the interesting things also that that we are very much studying right now with the SASP in these senescent cells that arise in vivo during regeneration is that later on, sort of during the late regeneration phase, they are starting to produce compounds that may help with their own clearance. Because when senescent cells are involved in a physiological process, be it development or regeneration, you probably don't want those cells that are producing all these pro-regenerative compounds forever. You need to eliminate them. And how do you eliminate them? Well, usually in these early systems, there is an involvement of the immune components. And we show that macrophages are important for that in salamanders, for example. And we believe they are attracted to this particular type of SASP that they are producing at later stages. So this is, again, something very interesting. And it's very interesting for two reasons. One, because it may explain how senescent cells are cleared at the end of regeneration, and then the process is perfect. So you start with nothing, you induce the senescent cells, you regenerate, and the final limb has no senescent cells. Now, obviously, if these cells are secreting, molecules that promote de-differentiation, you don't want them there once you differentiate your limb, right? So they are eliminated. But the other reason we're studying this relation between senescent cells and macrophages is because we had observed something which is very particular, and this is that salamanders do not accumulate senescent cells in any of the traditional structures where vertebrates, such as mammals, actually in humans, accumulate senescent cells throughout life. And we looked at salamanders, not only the younger salamanders that you usually use during your research, but up to 10 and up to 20 now years old. So up to 20 years old, that's, if to put this in context, the lifespan of an axolotl, say, is the reported lifespan is 13 years old. It is remarkably long for an organism of these characteristics anyway. But in our colony, so we have animals, which surpass 20 years old. So actually, when you give the animals the right conditions for living, and they don't have predators, they live happy. Sure. They have a lot of space because we are in Europe. There's very good regulations and very good animal welfare, laws and a consciousness to improve, the well-being of experimental animals, for example. And these animals really outlive their predicted lifespan. But if you take a salamander of 20 years old, you would consider it geriatric, super geriatric, and still no senescent cell accumulation. And why is this important? Because as you say, you had Judy here talking about senescence, and she must have mentioned to the audience that senescent cells accumulate in mammals, and an incredible amount of recent evidence suggests that there's a causal relationship between senescent cell accumulation and hallmarks of aging, and basically age-related disorders. And this gave rise, obviously, to this emphasis to finding compounds called senolytics, which eliminate senescent cells as potential treatments for improving health span, basically. Now, it's interesting that the salamander does exactly what we want to achieve in humans. They don't accumulate senescent cells. And so that's kind of one of the, probably the last branch of my lab is looking at these reasons of why salamanders do not accumulate senescent cells. And we're looking at several possibilities for this remarkable phenomenon, and not just the immune system, although the immune system is a very important component of this, but we're also looking at how salamanders regulate replicative senescence. Or how can they avoid telomere shortening and DNA damage? And we also are looking at the control of immune senescence in salamanders. So it's a lot of.
Yeah, I was saying. I'm thinking. Once again, you used the word marrying these systems, and it's a fascinating marriage that you're bringing all these together. I just want to while we're on the topic of the immune system, because you've talked about macrophages and you've talked about the clearing of senescent cells, talk for just a couple of minutes. In amphibians and some sort of, honestly, I mean, lower organisms, but as we go sort of down the evolutionary path there, their immune systems compared to ours, I mean, they have a much more active innate immune system than we do. Is that true? I seem to remember reading something there that there may also be, separate from sort of the clearance, that the innate immune system, although we have been obviously hearing a lot about it in recent years with COVID and sort of all the inflammatory problems that it's posed, in some of these organisms, it is their main sort of defense. And aren't there some interesting biomolecules as well that the innate immune response also can be pro-regenerative in certain contexts?
Yes, so I mean, as you mentioned, for sure, especially the innate immune response is involved in regeneration. Okay. That is for sure. And now, as to the particular molecules, in the case of the salamander, it's a bit too early to tell the characteristics, because what we know is that in the initial response to the loss of a limb, for example, these innate immunity marks that you find are different than what you find in mammals upon normal wounding. So you have a mix of M1 sort of pro-inflammatory and M2 more anti-inflammatory marks in these macrophages. So it's still a bit early to tell, but there's a lot of research efforts in those lines, especially scientists like James Godwin in the States are looking a little bit into this. But in terms of the actual characteristics and the potency of the different branches of the immune system in amphibians, it's not entirely clear. So yes, you can say they do have a nice innate response, but they do also have adaptive immune components. Okay. So, I mean, salamanders, and we're very much like interested in this, so they have T cells and B cells, right? The thing is, like, so far, no one has looked into this. Now, there's various reasons for that. If you think, when did we get the axolotl genome? Well, the axolotl genome was published in 2018. I mean, it's just too recent. And obviously, in order to understand certain processes, and particularly regarding the immune system, you need the tools, you need the genome, you need the transgenics to allow you to isolate the cells, to be able to characterize them. And that is happening right now. Mm-hmm. So I will be conservative and I won't address your question with a yes. No, because we do know that salamanders have these adaptive components too. And actually, and because we've been already talking about this in presentations in different international meetings, I can say this. So our lab recently discovered that that salamanders, in particularly the axolotl, can regenerate the whole thymus de novo. Mm-hmm. And the thymus is a key structure of the adaptive immune system. So it's the place of T cell maturation and the place where our immunity gets trained into recognizing self from non-self. And so it's an essential immunological organ for the adaptive immune cells. And one of the things we find is like they do have T cells. They have CD8, CD4 type T cells. They mature. There seem to be a lot of similarities between the process of T cell maturation in salamanders and mammals so far. Although, as I said, it's early days. We're characterizing the process and building the atlases, but I can tell you they do have an adaptive immunity. And there have been reports also of immune cells the adaptive branch, so T and B cells appearing in blastemas as they are regenerating. So whether they are doing something or not is an interesting question, really. So it's very interesting.
Max, so you've taken us on this journey into the world of regenerative biology and regeneration, of course. Talked a little bit about aging. I just want to go in one other direction with you while we have your time today, and that is into some other non-regenerative components of regenerative biology. And once again, we'll go back in history a bit, and I'm sure you're aware, and we've sort of touched a little bit about this in some previous episodes, but you know, in the regenerative biology literature, there's a bunch of those papers out there where, you know, we take our little salamander, we give them some nasty tumor, we chop off the arm, we get a blastema, we get a new arm, and mysteriously that cancer is no longer there. It's been normalized and made part of our new healthy limb. Are you doing anything interesting? And obviously these dynamics also are found in the plant kingdom, and you're well aware of that. Are you doing anything interesting with cancer? Is that something that, you know, is in the job jar, and we'll get a few more $1 million to your lab? What's happening in cancer regenerative biology?
The spot there, because it's something that first, I mean, it requires a lot of investment, and that's one of the things I should clarify. Like, salamander work is costly.
Understood.
But it's super interesting. Yeah. It's very, very interesting. And so some of this work, so obviously, like, these experiments have been done in the 50s and 60s. And it's just remarkable. And yes, as you say, like, you transplant a tumor within a regenerating structure, and that tumor either is not there anymore at the end of the process, or it gets incorporated potentially, or it may even induce axis duplication. But very rarely you get the tumor remaining. And this is also accompanied by a very striking resistance to tumor formation in salamanders, right? Especially, there's a lot of work by Panagiotis Sonis group in newts, hitting them with all sort of carcinogens and nothing happening, right? Yeah. So it's very difficult to induce in first place the tumors, and then what happens in regeneration, no one knows. Yeah. And I'm sure I'll be doing some things along the lines soon, and I'm sure some other people will be interested too. Now that we have the proper set of molecular tools, one can revisit all these very cool observations and experiments from the early 20th century even. Yeah. And it's wonderful. But as I say, all this type of detailed understanding and the detailed work on regenerating structures, or the connection between regeneration and cancer, or regeneration and aging, is really only possible to achieve and to pursue now that we have the tools that have for so long been available in other systems. But for us, I mean, this only became a reality a few years ago. Mm-hmm. So, I mean, now it is possible to tackle, but it's a very, very important topic, and also is connected with this very interesting thought that came especially from the mammalian wounding field, always comparing wounding, cancer, regeneration, and cancer, and saying, well, maybe we lost somehow the ability to regenerate because it may sort of promote cancer formation. But what these animals that regenerate that well tell you is that there are ways of doing it while being cancer resistant. Mm-hmm. Mm-hmm. So this is a very interesting contribution from the salamanders, I would say.
Yep. I take my hat off to you and them.
They deserve all the credit, I would say.
Max, while we still have you, we talked about the differentiation and differentiated state, the cellular plasticity. We talked about senescence, the immune system. Any other things that you want to highlight while we have you? Any other programs, things that I've missed? I want to make sure that I give you enough time to talk about anything else going on at your amazing lab.
I mean, one of the things that we are interested in, which is in connection, with this remarkable sort of longevity and lack of aging signs that we find in salamander, in the salamanders in general, is the connection between these amazing regenerative abilities and the control of biological time. So you probably heard from the recent work of people such as David Sinclair that look at reprogramming and actually suggest that there might be a connection, a resetting, at least of the epigenetic age of cells during reprogramming events.
Sure.
So this is something we're very interested in, I think, because I think it's very easy to say, well, an organism that regenerates everything, of course, is not going to have all these aging signs because it's regenerating constantly, but that organism is aging at the same time. So it's interesting to see what's the interplay between regeneration and aging, and particularly, how regeneration of an old structure impacts on the age of the cells involved, for example, or the overall physiological age of the animal or the age of other tissues. And this is something that is important, and we are also very interested in, and I'm sure other people will be interested in as well. So that's a very important point. And one final, if I may,
sure,
factor that I will stress that is being pursued in the field much more now is the effect of systemic signals and how systemic signals may impact regeneration. So that's another sort of important area that is active in the field right now. So look at regeneration not just in isolation, but as part of the organism. And we know that that signaling, systemic signaling, can really affect regeneration. I mean, there's, at least while these studies have not been done extensively in salamanders yet, there are cases in the regeneration of the imaginal disc of Drosophila, for example.
Okay.
So the imaginal disc of Drosophila, as Drosophila goes through these larval stages, it can regenerate. It can regenerate very well. So let's think of the wing disc, the disc that is going to eventually give you the wings of a fly.
Mm-hmm.
So someone can resect within the larvae that disc, and it will regenerate. Now, that is able to be controlled. That can control, for example, the time of pupation of the fly. So if you don't touch the disc, the fly forms the pupae at a certain time. If you induce regeneration, then that fly will pupate much later, allowing time for regeneration. So there is a connection between regeneration and the rest of the organism. And that is important. And there might be other aspects of the organism physiology that impact on regeneration and how well this process proceeds.
It's, as said at the beginning, it's definitely something that I have been amazed by, but listening to thought leaders and experts like yourself, I really, if I had a hat on, I'd take it off to you. I'm really wishing you the best with all this, for everybody listening, and for all funders of this type of work. I mean, if there's any lab that I could point to that is going to be doing the most cutting edge work, Dr. Maximina Yoon, I mean, this has really been eye-opening, and I'm really wishing you the best with all of this. I'm going to be definitely staying in touch, and I would love to do a follow-up episode in terms of these other concepts that you brought up during the call in terms of positional identity, and really a reawakening and a reconnection of these themes in 2022 is just so fascinating. For everybody that is going to be listening to this episode across the podcast networks or watching on the YouTube channel, once again, you've been listening to the amazing Dr. Maximina Yoon, Research Group Leader, Center for Regenerative Therapies, Dresden, Technical University of Dresden, jointly affiliated Max Planck Institute for Molecular Cell Biology and Genetics. Max, again, thank you for taking the time out of your schedule, long time, to talk to us about all these topics. Obviously, thank you for everything you're doing there. And as we like to say on this show, thank you for helping to create a better tomorrow through your research. It is such a fascinating program you have.
Well, thanks to you, Ira, and I would love to be back for another episode. And I hope the listeners enjoy this and get into regenerative research and perhaps into the salamanders.
Absolutely. Great seeing you.